Mhm.
Okay, so could you introduce yourself to our audience?
Definitely. My name's Robert Mayall. I'm one of the co-founders and the Chief Technology Officer at FREDsense Technologies. We are a Calgary, Canada-based startup company focused on developing new synthetic biology-powered ways of monitoring what's in the world around us in our water.
So what is actually the story behind how FREDsense started? And how were you inspired to sort of pursue water quality monitoring water quality monitoring?
So back in 2011 and 2012 at the University of Calgary, myself and my co-founders were part of our university's iGEM team. And we really wanted to do something local for us. And in our province in Canada, there's a lot of oil extraction processes, and they generate a lot of used water, referred to as process water, in their extraction systems. We wanted to find ways to monitor for any potential toxins in that water so that the water would always be safe if it were to be released back into the environment. What we really came across while doing this, though, was a platform for getting biological systems to send electrical signals to whatever recording device you have it hooked up to. It could be as simple as a Raspberry Pi, it could be a complex integrated circuit, whatever electrical system you're looking at. During the 2 years that we were in iGEM in 2011 and 2012, we had an absolute blast. We built up a large team around us and we did quite well at those competitions. But then after the 2012 Jamboree, I remember us sitting down and saying, well, what now? We don't want this to stop. We'd had so much fun doing this and we felt that this was such an important avenue to be working on, so something that could have a real impact in the world. And somewhat naively at the time, we figured that the best way to do that would be to start a business. So we joined the entrepreneurial track at the time for 2013 and started trying to assemble advisors and mentors to teach us how to run a business because we were a group of science students at the time. And then after that competition in 2013, we incorporated as a business in 2014 and have been going since then.
So is it would it be, so when you mentioned that you're sort of having sort of almost like a platform for this sort of electrical signal sensing, are you primarily using that for water quality monitoring, or do you have sort of, are you branched out into different sort of applications?
So we focus on having these electrical outputs from biological systems. And we do that for two main reasons. The first is that we've seen in multiple applications that it can be more sensitive than typical GFP luciferase type measurements. So that is a huge benefit when you're looking at trace chemical detection in a water sample. The other thing that's really useful about this, though, is the electrical output doesn't care about the turbidity of a sample. So if you've got a sample that's got mud or suspended solids in it, or in some of those oil applications that we looked at back in the beginning, could quite literally have oil slicks on top of them. Light-based outputs don't really work in those, whereas the electrical output loves it. In fact, in those process water samples that we looked at originally, they were quite salty, which made them very conductive. And while fluorescence-based measurements didn't work at all, our system was better than our initial testing because of that conductivity.
How is I'm just wondering, how is currently water quality like monitored like by companies? Yeah, generally.
So there's generally two different ways that people go about measuring water quality right now. The first is to use big analytical labs, things like an ICP mass spec or HPLC mass spec. These are phenomenal tools, but you need to have a very well-trained technician to run them, and they're very expensive too. On top of that, they're usually off-site as well. So you have to have somebody going around taking samples, and then you have to ship them to an offsite lab, fit into their queues, and then get your results back at sometimes quite a delayed, I think the longest we've seen so far is 4-week turnarounds. So if you are looking at cleaning something and optimizing a remediation process, for example, that means you could be optimizing the process with data that's 2, 3, 4 weeks out of date. The other way that people go about measuring water quality right now is very quick field tests. These can be as simple as dipping a strip into a water sample, just like a pH test or a litmus test. And there are some that are a little more complex. For arsenic, for example, there's one where you generate arsine gas and get it to react with mercury on a piece of paper. These tests are usually really quick, 15 minutes or less, and they're almost always done on site by somebody who does not have a significant degree of chemistry training, because you don't need it to run these type of tests. The downside is that while they're much quicker and easier than an analytical lab, they're usually far less accurate. For arsenic, for example, there's one test that looks at bucketing the concentration 0 to 10, 10 to 30, 30 plus parts per billion. When you're trying to optimize a process, having a range of plus or minus 20 part per billion can make it quite challenging to tune your system.
And so by using bacteria, you can, those synthetic biology tools, you can provide sort of a faster system that is also more about the real-time sensing.
Yeah, so at FREDsense, that's the avenue we've taken. So you kind of have these quick field tests or these harder to use analytical lab tests, and we're in the middle of those. So we have engineered bacteria to do the heavy lifting of this detection for us, and they will sense what's in the world around them and then send us that electrical signal. It's a lot simpler to use than some of these tests in that you add the water sample to the bacteria, give them a good shake, and then plug them into our devices. From there, the bacteria do everything. However, it is slower than some of these quick dip a strip into the water sample tests, because the bacteria have to actually react and then produce the response. So with this, typically within 2 hours, sometimes within an hour, we can produce responses that are down to plus or minus about 2 parts per billion. So significantly more sensitive than some of these strip-based tests, and a lot faster than an analytical lab would be for this.
And what have been sort of in producing these systems, what have been some of the major challenges that you've come across?
Yeah, I would say there's probably 2 major challenges that we had to overcome in getting this system out into the field. The first was shelf life. When we're synthetic biologists in a lab and you want to do some kind of incubation or an induction experiment, you take a glycerol stock, make an overnight. The day of the experiment, you'd make a subculture, and then you would start after it had reached whatever OD was appropriate for your experiment. That doesn't work in the field. End users typically don't have -80s with glycerol stocks and can't set up an overnight and then a subculture and then you expect to find the right OD. So not only did we have to find a way to preserve our bacteria for months at a time, because they're not going to want to order it and receive it the day of, they need to have a stockpile of these tests ready to go. But we had to make sure that as soon as it was ready to be used and the water sample was introduced to the bacteria, that they were ready to go right away. They didn't have to grow for 2 hours and then have the water sample. So that was one significant challenge that we had to face there. The second thing is, while as scientists, we're quite used to how to set up a lot of these experiments, moving microliter volumes between multiple different tubes, having our own shorthand on top of the lids of these tubes, these are actually relatively finicky tasks that we've all learned to do. And that isn't a skill set that most of our end users have. So something that we had to do was translate all of the lab-based science into an easy-to-use format that could be as simple as add water to this chamber and shake, and then plug it in. And get the bacteria or the device itself to automate the rest of that.
Is your are these sort of kits, field kits, are they each customizable based on what they are sensing?
Yeah, so that's really the underlying thesis of FREDsense. We have a platform technology for getting responses from biological systems, bacteria and otherwise, about what they're seeing in the world around them. And with synthetic biology and the tools that we have in this field, we can tune that to be multiple different parameters. The first commercial sensor that we've released is for arsenic monitoring, but we do have multiple others in the works.
What other sort of chemicals are you sort of considering sort of monitoring? What are the ones in the works?
Yeah, so we have a handful, but the ones that are furthest along and that we've made publicly known are different metals that could be in groundwater samples that occur in a similar area to where you would have arsenic contamination. So we've got things like iron, manganese, zinc, things like that.
Why would you think it is important to take water quality like assessments regularly, and how little do we know about how accurate this can be?
Sorry, what do you mean by that?
I mean, like, how little like do we know about how accurate the water quality processes can be? Like, when one analyzes the water qualities in like facilities, are there like any challenges with that come around besides taking quite a long time and more specialized like labs, for instance?
So it's pretty context dependent. Depending on which market you're looking at, which analyte you're looking at, there's different rationales. On the arsenic side, for example, which being our first commercial sensor we've got the most experience with, one of the strong use cases for a technology like this is in remediation systems. So what I mentioned before, you could be optimizing a remediation process with data that's quite out of date. Now, there's a lot of places, especially in, say, the southern US, where there is arsenic in the groundwater that they use there. And all of these cities, they've got big systems set up to remediate this arsenic and to remove it from that groundwater before it's pumped into people's homes. But to make sure that they're always keeping this water safe, they will take those readings and then make sure that they're over remediating unless that data is very recent. So that means an increase in costs, as to remediate, you need to spend money on different chemicals to remove that arsenic. By providing faster yet still accurate data, it allows these operators to tune their processes more, so that instead of spending more money than is needed, they can remediate to just the right level when they need to do that.
And does the use of your field kits differ between if that you're using them in different sort of water sources? So like if you're monitoring at groundwater versus, let's say, wastewater, if you had to monitor in wastewater, or lakes and rivers, or is it only suited to one particular water source?
That's a really good question. We've tested multiple different sources, surface water, groundwater, where surface water is lakes, rivers, things like that, as well as wastewater. For the arsenic system, we've mostly focused on groundwater, but we do have other systems that have looked at, especially wastewater. Something that we started doing during the pandemic was doing monitoring for COVID-19 in wastewater. We've really found that our expertise is on getting lab-based systems in an easy-to-use format. So we applied that for wastewater-based epidemiology, and that has needed the use of wastewater for that.
Mhm.
And I'm wondering, is there any challenges that your company has had to face in the past regarding using synthetic biology to like monitor water quality? Like in maybe like in the public perception of like using engineer organisms for these purposes.
Yeah, and that's something that when we started, we weren't sure how to go about doing this. Especially in iGEM, for example, it's drilled into you that you need to make sure that you're always being conscious of what could happen to the environment should your organism be released or escape in some capacity. So we've actually had lots of work done with different regulatory bodies in the US and Canada, and our organism has actually been approved for use in our cartridges in both countries. So we did have to go through a process to make sure that we had regulatory approval for this.
How long did I'm just curious, how long did that process of approval actually take?
Oh, that's testing my memory. I believe that took us about 12 to 18 months. It was not a quick process, and we did have to do a lot of extra tests and provide a significant amount of data.
Yeah, also wondering how exactly, how could you explain more in detail how does the synthetic biology mechanism work for like sensing water, doing like water quality like monitoring?
Yeah, definitely. I can give a bit of an overview on more of the synbio aspects of our system there. So we've got quite a few different platforms now. We can work with DNA, RNA detection, we can work with protein-based detection, and we can do more whole cell biosensors. And with whole cells being more of the traditional synbio approach, that's the one I'll focus on for this. The simplest system that we've used on this is the old school promoter turns on a gene type system. So we have screened many different promoter, RBS, transcription factor type combinations internally. And we've also recently been starting to explore partnerships with other groups that can do this type of screening for us. Just at the Built with Biology conference, we announced that we've signed a partnership with Ginkgo Bioworks, where they're developing a new suite of sensors for us. So with these, the molecule of interest will enter the bacteria that has been engineered, trigger the transcription of a reporter gene. And for us, that gene is a proprietary one that catalyzes an electrochemical reaction. And we can detect that reaction electrically, which is converted to a concentration automatically with our hardware systems.
Okay. And when you were like starting, can you like tell us more about how it was for, in your case, like the transition between like an iGEM startup and an iGEM project to like a startup, and what were like one of some challenges that your company had to face when becoming like a biotech startup in the States and Canada?
Yeah, I will be completely transparent. It was tough to start. We were a group of scientists who, when we started the business, we did not have strong business backgrounds. Thankfully, two of my co-founders, Emily and David, they have had an interest in business far longer than I had. So they knew a lot more upfront than I did. But even then, we didn't have MBAs, we didn't have previous experience with companies. One of the things that we did, though, right at the start, was we applied for every business plan competition and business pitch competition that we could find. And through that, we actually found a lot of structure in these competitions. Things like you had to have one slide describing your target market, one slide describing your manufacturing. And what was a real shock to us at the start is the technology was usually one slide out of 20. And being a group of scientists, that hurt a little bit at first. But having that forced on us, that the technology is one small aspect of a business, really helped transition our mindsets. From there, we were lucky enough to get accepted into the Singularity University's first accelerator cohort. For that, David and Emily went down to the Bay Area and spent a few months down there learning. And they were able to make so many connections with phenomenal mentors. Those who've been in the biotech space, in the startup space, some in the biotech startup space, and really build out a network that they'd been able to learn from. That unlocked so much more for us. Having a good group of mentors is something I cannot emphasize enough. Without that, it would have been nigh impossible for us to have developed as far as we did.
What about in terms of IP? Because as part of the iGEM competition, everything has to be publicized, of course. So how did you tackle sort of challenges surrounding intellectual property?
Yeah, IP in iGEM can be a bit of a challenge. I don't know what the laws are in the UK, but in Canada and the US, from the moment that you disclose an innovation, you have 1 year to submit a patent application on it. So assuming that the iGEM competition is the first time that you disclose your intellectual property, you can patent it up to 1 year later than that. At least in the US and Canada.
Yeah.
I'm not sure if that's the case in the EU and the UK.
I think for us, it's as soon as it's public property. So you are not allowed to patent public property. I think that's the case here.
Though I believe you could submit a provisional patent application prior to the competition, if that was your goal.
Yeah. Probably, yeah.
Also keep in mind that there are companies that have been quite successful without relying on a closed source model. One example of that is Chai Biosciences, the Open PCR and Open qPCR group. All of their software and designs are available online, and they have quite a robust product that multiple people have purchased.
Yeah. I actually have like one more question about the water quality monitoring, and yeah, I'm just wondering what do you think is next for like water quality monitoring with synthetic biology? Like, is your company or the whole area like facing some certain challenges that you think need to be addressed like in the near future?
I think what's next for water quality sensing is going to be in making bigger and broader panels in the testing systems. So instead of having an arsenic test, a lead test, you have a suite of tests that could all be run at the same time. How can you make big panels like that? Similar to how if you get blood work done at a hospital, they don't typically test for just one thing. They'll test for multiple things at once. I think taking that mindset and applying it to water quality monitoring is going to be quite revolutionary. Right now, a lot of times that means buying different systems for different analytes, which is a lot of upfront investments. That's one of the big reasons at FREDsense that we've always taken this approach of, we have a hardware system and you could plug different tests into that. So an arsenic cartridge goes into the detector, a lead cartridge goes into the detector, and that one piece of hardware could do all of them. So that end users don't have to buy 10 different detectors. It's just, you have a testing unit and you buy whichever tests you need that day.
And how do you see sort of the next steps for your particular platform? What, aside from sort of going into different, sensing different contaminants, what else do you have sort of in store in the next few years?
Yeah, so as well as broadening our current contaminant detection library, so to speak, something that I'm really excited about is expanding beyond things like trace metal detection. So with our recent work on detecting COVID, we've branched into DNA and RNA. How far can we extend the different breadth of things that we can sense? Could we do in a single box? Could we do DNA, RNA, proteins, fatty acids, carbohydrates, metals, inorganics, everything? That's what I want to create. I want to create the system that allows you to know what's in your water, no matter what it is that you want to know.
Yeah, I think
there's actually, no, there's one question I want to ask, but I'm not sure quite how to ask it. because of course, sort of when have been sort of reading into the literature of sort of monitoring water quality and sort of taking water samples and things like this, sampling water, I found often that, you know, lots of little samples have to be taken, and the process, you know, you sometimes wonder how, no matter the accuracy of the tests, how accurate is the monitoring because of where you locate your sample taking. And I wonder sort of whether FREDsense has thought about, let's say, upscaling in some ways the field tests, or sort of have thought about this kind of challenge in water quality assessments.
Yeah, that's a really good and also a really specific question. So with this, it depends on what the application is for. So if you're optimizing a treatment process, for example, you're going to have a pipe that comes in and a pipe that goes out. So in that situation, where you sample is quite obvious. But for a lot of other applications, especially things like surface water monitoring, you're right that where you sample, if you're looking at a trace contaminant, it may not be in that part. So one of the things that people will do is referred to as plume mapping. So that is going all the way around a perimeter of a spill, a lake, whatever water is of interest, and try and figure out where the contaminant is and where it isn't, which can be just as important. So that is a really good application of figuring out where you sample. Another application of that that's really important is actually in COVID-19 wastewater detection. So you can imagine if you've got a city or a town, there's thousands upon thousands of toilets all feeding into the wastewater system. Depending on where you monitor, be it at a building, at a manhole, at the wastewater treatment plant, you will get different answers. And you can actually trace back to which buildings, which communities, which wastewater treatment plants have different levels of COVID-19. And at least the theory that has been discussed in academic papers is perhaps tailoring the public health responses on a more localized level. So there's a lot of really interesting applications there.
How about tracing back like the antibiotics maybe from like animal runoff and stuff like that? Can your system like already detect this where the maybe like antibiotics are running to the water, or do you think this can be done maybe in the future?
Yeah, detecting the spread of antibiotics through a watershed could probably be done the exact same way through something like this. Though you'd most likely need to look at breakdown products from those antibiotics, in the same way that in a lab you're taught don't leave the antibiotics out at room temperature in the light all the time. I doubt they would still be in their initial form if they've been through an animal, excreted out, and then spent some time in a watershed as well.